welcome to Intro 101. My name is Chris, and I am your Neuroscience and Psychology Graduate Student Assistant. I'm here to assist you with the related science basics that you interact with on a daily basis, much like a graduate assistant would do in academic settings to help prime students with introductory level instruction so that they're ready for lectures given by their professors. Now, in the last week, there has been some controversy in women's boxing at the Paris 2024 Olympics. The brief bout featuring Iman Khalif and Angela Carini has sparked masses to jump to conclusions about Khalif and her anatomy and biology. I'm not here to weigh in on her anatomy or biology because it's none of my business, and I'm not qualified to comment on the IOC or sports in general. I'm here to share excerpts of episodes I've posted that give a bit of a primer on chromosomal and genetic anomalies that affect gonadal development and how that can impact sex-specific hormone development. They're not directly related to this particular controversy, but you might find them helpful to learn that chromosomal and genetic anomalies are a lot more common than you think. Sports medicine doctor and YouTuber Dr. Brian Sutterer posted a wonderfully balanced perspective and overview of the many variations of chromosomal and genetic anomalies that should be considered before drawing any conclusions about Iman Khalif or anyone ever. I'll post a link to his video below. The excerpts I've included here will hopefully add some context to the information he gives in his video. I'll post a link to the full episodes I've pulled these excerpts from if you'd like to learn more about brain and body physiology and development. As always, thank you for watching and take care out there. Okay, next we'll look at the nucleus and then get to the non-membranous organelles afterward. Most of them will make more sense after we discuss the nucleus anyway. Okay, so the nucleus is the largest membranous beast inside the cell universe, but it's not an organelle. It's its own entity, usually located somewhere near the middle of the cell. It is where your deoxyribonucleic acid lives. You'll know it as DNA, and it is all of your genetic information, and it's neatly packaged into chromosomes with other nucleic proteins. Each nucleus in most human bodies has 23 pairs of chromosomes. That's 22 pairs of autosomes and 1 pair of sex chromosomes. There are some instances when humans can have chromosomal anomalies. These anomalies are organized by either numerical anomalies or structural anomalies. Structural chromosomal anomalies include deletions or duplications to parts of the chromosome, and these affect the amount of genetic material in the cell. Numerical chromosomal anomalies are when someone has an extra chromosome or is missing a chromosome. This is called an aneuploidy. A common aneuploidy is a trisomy, and a trisomy is having a third copy of a particular chromosome. Down's syndrome is one example of a trisomy to chromosome 21. So people with Down syndrome have a third copy of chromosome 21 in each cell instead of just the pair. Klinefelter syndrome is another example of a trisomy, but this affects the sex chromosomes. Klinefelter syndrome is when someone born with an XY chromosomal pair has an extra X chromosome, so their chromosome makeup would read XXY. It is also possible for XY humans to have an extra Y chromosome, and this is called 47XYY syndrome or Jacobs syndrome. Another example of an aneuploidy is a monosomy, and that is the loss of a chromosome in each cell. An example of a monosomy is a Turner syndrome. People who are typically born with XX chromosomes but have Turner syndrome will only have one copy of the X chromosome in each cell instead of the typical XX chromosomal pair. There are many types of aneuploidies, but only a few of them are compatible with life. And those that are will have differing impacts on developmental and or reproductive outcomes depending on the genetic features of the chromosome affected. Because I'd like to devote the remaining minutes of this episode to the genetic and hormonal influence on gonadal differentiation and how that may influence brain sex differentiation. And this is just a fancy way of saying that depending on the embryo's gonads, some of its brain structures will later undergo masculinization or feminization. And those are phenotypical labels given by scientists to distinguish the structural differences between brains that marinated in androgens in utero and brains that developed in the absence of androgen exposure. But I want to be clear-ish about a couple things up front. One is that the word differentiation in this instance is different from how it's been used up to this point in the episode. This is not about pluripotent or multipotent stem cells morphing into a particular type of cell within one individual. This is about two phenotypically different individuals and the differences between their brain development. The second is that sometimes brain differences are phenotypically distinct, and sometimes they're not. Sometimes there's a lot of overlap and/or small differences between them. From what I can discern from the literature, discussions on brain sex differentiation are based on averages. So to understand what I mean by brain sex differentiation, we have to go all the way back to the beginning of this episode when the sperm cell dropped its haploid nucleus into the oocyte. Remember, that's half of a full chromosomal set. So that sperm haploid only had 22 autosomes and one sex chromosome, either an X chromosome or a Y chromosome. And the oocyte had 22 autosomes and only an X sex chromosome. If the sperm haploid had a Y sex chromosome in its chromosomal set, the resulting zygote would have 44 autosomes plus XY sex chromosomes and will go on to produce testes. If the sperm haploid had an X sex chromosome in its chromosomal set, the resulting zygote would have 44 autosomes plus XX sex chromosomes and would go on to produce ovaries. So typically, to be clear, XY zygotes will go on to grow testes and XX zygotes will go on to grow ovaries. Now there are chromosomal anomalies that can happen, and I explained what some of those are in the cell physiology episode, so I won't go over them again here. But as the zygote matures, whichever set of sex chromosomes the embryo has will determine the embryo's reproductive organs, and those organs may or may not secrete certain hormones that will later partially influence the morphology of some brain structures. So, if our embryo's reproductive organs are testes, it will produce androgens and secrete them into circulation, and those androgens will inform how some of those brain structures will form. If its reproductive organs are ovaries, there will be an absence of androgens, and that absence will inform how some brain structures will form. There are also transcription factors thought to be involved. It's likely not just androgens or the absence of androgens that induces differences between XX and XY reproductive organs and brains. But androgenic influence has been the widely accepted understanding of brain sex differentiation. But let's take a closer look at what's happening. As the embryo develops its reproductive organs, the gonads are bipotential, meaning until they are told to become testes or ovaries, the developing reproductive organs are virtually the same, regardless of XX or XY chromosomal designation. There is a very sensitive window of time during gonadagenesis when sex determination happens. What turns on gonadal determination is either the presence or absence of a gene called SRY. SRY is present in the Y chromosome, and its expression encodes a protein called testis determining factor, or TDF. And TDF kicks off the differentiation of steroidogenic precursor cells. Those are going to produce the androgens in this instance. They also kick off primordial germ cells in the developing gonads, which differentiate into those hardworking Sertoli and Leydig cells, and some of those will differentiate into cells that form the testes. The fetal Leydig cells, those are going to produce androgens and pump them out in abundance. Those androgens will continue to inform the development of the body and then later the brain. In particular, there is a surge of androgens which include testosterone and dihydrotestosterone, or DHT, and those influence the development of genitalia as well as the Wolffian duct that will differentiate into the epididymis, vas deferens, and seminal vesicles. The Sertoli cells secrete anti-Mullerian hormone, which represses the development of the Mullerian duct. The Mullerian duct is the precursor to the XX reproductive tract, which includes the fallopian tubes, uterus, cervix, and the upper vagina. Its regression also allows for the formation of the Wolffian duct. If the embryo does not have a Y chromosome and instead has two X chromosomes, it won't develop testes. There may be some transcription factors in XX genes that actively repress testes formation, but it's not yet certain. It's mostly been thought that if the SRY gene doesn't launch testis-determining factor, then the reproductive organs will just passively become ovaries. So we pretty much don't know entirely how ovaries become ovaries outside of the absence of androgens. And I'll say it now, and remind you again later, we don't seem to know much about the mechanisms that feminize brains either. And ovaries also secrete sex-specific hormones, but not until puberty. So the current understanding is that without exposure to androgens or anti-Mullerian hormone, the ovaries will initiate development and the Mullerian duct will continue to develop because there's nothing stopping it, and it begets most of the XX reproductive organs. Now, gonadal determination occurs around the 6th week of gestation, but brain masculinization or feminization doesn't occur until the last trimester, so beginning somewhere around the 25th week. Until then, the brain in both XX and XY embryos are also bipotential, meaning they can be either masculinized or feminized. And again, all that really means is that some brain areas organize slightly or not so slightly differently based on the presence or absence of prenatal androgen. And though masculinized brains can show childhood proclivity to objects and playstyles and behavior, socially deemed masculine, the theory is that many of these brain differences don't fully manifest until they're activated at puberty, when either testosterone influences the XY brain or estrogen influences the XX brain. Though that theory has mostly been tested in rodents and birds that sing. There are other brain sex differentiation theories involving XX and XY chromosomes and their gene expression. For instance, the SRY gene in the Y chromosome may continue to influence brain differentiation later in gestation, and both chromosomal sets may have genes that encode transcription factors that influence masculinization or feminization of some brain structures. But we don't really know, and that's less to do with hormones, so probably a topic for another day. The distinctions I want to emphasize are first that gonadal determination has a very sensitive window. Any disruption in androgen exposure in XY embryos will kick off a cascade of effects that will affect gonadal differentiation and possibly later brain masculinization, though other transcription factors may offset some of those deficits. We just don't know. Conversely, exposure to androgens in XX embryos during that window also kick off a cascade of effects that impact gonadal development and possibly later both brain masculinization and feminization, or neither. And this is where we close the loop on Dr. Ben Barris's story from the Glia episode. Dr. Barris's mom was prescribed an androgen-based anti-miscarriage drugs during her first trimester, which would have passed into fetal circulation through the placenta, exposing XX embryonic Ben to androgens. Ben was born without most of his reproductive tract, though he did develop ovaries. He also suffered from gender dysphoria from as early as he could remember until he transitioned to his experienced gender at midlife. And being the consummate neuroscientist that he was curious if his early embryonic exposure to androgens caused a developmental cascade that led to the masculinization of his brain, and perhaps partially explained his proclivity toward masculine-labeled toys and interests throughout his childhood. Though he was quick to acknowledge that neither does one have to be prenatally exposed to androgens to experience gender dysphoria, nor does one require brain masculinization to have an interest in socially deemed masculine playstyles and objects. I mention it because he is an example of an XX embryo who was exposed to androgens during the sensitive gonadal determination window, which may have affected his reproductive tract development and possibly influenced a masculinization cascade. The second distinction I want to emphasize is that gonadal determination is not concurrent with brain masculinization or feminization. There are many weeks and a lot of brain development separating these two processes, meaning any disruption to neurodevelopment in between those events could affect the process of brain masculinization or feminization in the last trimester. There are some theories that suggest disruption to brain masculinization or feminization may be correlated with gender dysphoria and or gender fluidity. This is difficult to disentangle until we have a better understanding of the mechanisms of brain masculinization and feminization, as well as gender dysphoria. But it has me curious if there is a connection between disrupted prenatal neurodevelopment and atypical brain sex differentiation, and how that might influence gender identity. Okay, lovelies, thank you for being here. Again, please check out the STEM organizations in the show notes and support them or others in any way that you are able. I have no relationship with these organizations. I'm just excited about the work they're doing, and I hope that you will be too. If you liked this episode, please click the like and subscribe and notification bell if you haven't already. Please share it with your friends, neighbors, and family, and anyone who you think would benefit from understanding their body a little better. Until next time, take care out there.